Hey, Bronwyn,
Hmm?
what's the chair for?
We're having a visitor.
We're having a visitor?
Yes.
Awesome. Who is it?
It's Nick Harvey.
Nick?
Yeah.
I did my honours with him.
Hi guys.
Hi.
Hi,
I'm Bronwyn,
I'm Nick.
I'm Aaron.
and we're here to talk to you today about Week 10's prac.
Glucose tolerance test. As the resident diabetic of this trio, I'm actually familiar with this. It's where you have, as a potential diabetic, it's a test where they get you to have a glucose drink and they take your blood at 0, 1 hour, 2 hours, and, kind of see if your levels, go up and then correct back down to normal. And if they don't, possibly a diabetic.
So we're doing this glucose tolerance test today in 2 different methods. So the first test today will be utilising glucose oxidase and peroxidase to determine how much glucose is present and to produce a colour so that you guys can measure it in the spectrophotometer.
The second method is an electrochemical method using a glucometer, and you'll be taking a little bit of the patient blood and the electronic chip, just putting it in and testing it. Nice and simple.
Yeah, sounds easy.
It is. I actually do it all the time.
So yeah, I found something interesting today about today's prac.
Yeah?
The first enzyme that we'll be using, glucose oxidase, isn't present in the human body, which of course you don't really want any of these present in the human body because then it's breaking down the things that you need into forms that you don't want, like gluconic acid, nasty stuff if you have it in your body. But it is in fungi and in insects, and there glucose oxidase is used to create the H2O2, hydrogen peroxide, that we will be creating in the prac today as well, but they use it for killing bacteria.
I don't get it. Without glucose, how can they still be a fun guy?
So obviously when the hydrogen peroxide is generated, then we need the second enzyme to do that colour development. So you need the peroxidase?
Yep. And then we create, using the peroxidase, we convert the hydrogen peroxide. It reacts with 4-aminophenazone and with phenol and creates quinonamine dye. And that's what we're measuring in the test.
It's a pinky colour, isn't it?
It is. And we'll show a picture of that later.
Yep, cool.
So we're using Jack and Jill as our patients, and so of course you'll see a change in one and not in the other.
Well, you might. They might both be the same.
They could be.
Yeah.
You never know.
The plot thickens.
Are they both?
So-
Are they potentially both diabetic? Are they potentially both normal samples?
So this is where our hypothesis will come in, guys.
So what would you think a good hypothesis could be for today's prac without giving too much away? Hints.
Yep, so you can't say that one will have diabetes and one won't because we don't know yet. So what you can hypothesize is that if their glucose is outside of the normal range at the 2-hour mark, then you will say there is a possibility that one of them has diabetes.
So Aaron, do you think there's any other way we can do this hypothesis?
Well, we're actually testing 2 different methods. So is one, possibly more reliable than the other? Is there potential errors being introduced?
So say we were to go down the reliability track. Do you think that we can prove that from this test?
We do have a quality control.
We do.
So
They do.
they do.
So we can check the accuracy.
So, I don't know, one method might be a little bit more accurate or reliable than the other, whereas the other might be just more expedient though. Alright, so the method for the glucose oxidase. You're obviously going to have to do your standard, your quality control, your blank, and your 6 samples based on the 2 different patients.
So
9.
patient 1 or Jack
Yep.
will have 3 tubes,
And Jill
Jill
will
will have 3.
Yep. Little video of what to do is you take 10 microliters of the patient's serum sample put that into your tube, followed by 1,000 microliters or 1 milliliter of the glucose oxidase assay reagent. Just to point out, the assay reagent actually does contain a lot of different things. It does have your 2 enzymes in it, it has phenol in it, and it's also got the 4-aminophenazone.
So that makes it a lot simpler for them. They don't
It
have
does.
anywhere near the pipetting to do that they might have had to.
Yeah. So instead of having to do this reaction in 2 separate steps, you can do it all in one because
Everything's in together.
Yeah, everything's in together. The enzymes are highly specific, so they're not going to react with any of the other, reagents mixing around in there.
And then you will mix it well and then incubate it at 37 degrees for 10 minutes.
Which will produce your colour change ready to measure on the spectrophotometer at...
500 nanometers, because that's the wavelength that pink comes up at. Right, now guys, we're just going to give you a quick look at some pictures from an experiment that we've done. So you can see your blank sample is clear because it doesn't have any glucose in it. Your quality control sample is slightly pink, and your standard sample is a really nice pink color, so you know the concentration of glucose in that tube.
Here we have the time course for Jack. And the time course for Jill. So, Nick, using the glucometers, step us through what we're going to do.
Sure. So firstly, you're going to get 6 microliters of your blood and make sure it's well shaken before you pipette it out. You're going to get a strip of parafilm and just drop your blood sample onto the parafilm. Then you're going to get the chip that goes into the glucometer. You're going to put it in and wait for the glucometer to flash up with numbers, and then just touch the end of the chip to the blood spot, and you should get an accurate reading from that.
Awesome. Nice and easy.
So this is a simple prac.
This is a very simple prac.
And I don't think you guys should have any trouble with the calculations today.
Nice and easy.
Yep.
And just to reiterate, when you're doing your discussion and questions, keep in mind what Aaron said at the start and why we're doing this test and what the results are.
Yep.
And have a great prac. We'll see you in there.
Bye.
Bye. So Nick, while we've got you here, we thought we should ask you a little bit about yourself.
Sure thing. What would you like to know?
So, did you do your degree here at QUT?
I did, yes. I did applied science when it was still a thing, but
Mhm.
I think they've changed it to just Bachelor of Science now.
Okay. And so you did honours,
Yep,
and now you're doing a PhD.
I am.
So your PhD is pretty special, isn't it?
I think so, yeah. So what happened was when I finished my honours, I was really questioning what I wanted to do because medicine's still in the back of my mind. And then was searching around for a PhD project that would give me a lot of fundamental techniques that I can pretty much take anywhere in science. So that's why my project includes a whole bunch of DNA sequencing, some epigenetics work, some transcriptomics, basically all the 3 fundamental things that you need for successful research projects nowadays.
Okay.
And biochem help you out at all?
It did in a big way. Pipetting schedules are a thing, guys. You have to know what you're adding, you need to know when you're adding it. They're very, very helpful for organization.
Okay. Personally, in my PhD, I'm jumping between chemistry and biology and nanotech, and I'm all over the place personally. But the fundamentals that I've learnt in biochem personally have been of great help. Even if it's, for simply understanding, PCR, because it's an enzyme reaction and what it's actually doing. And yes, we're talking molecular biology, but the underpinning fundamentals seem to be across all the sciences. So
Yeah, and multi-omics approaches and multi-team sort of development is where everything's coming to. So I hope you guys keep in mind that you need a pretty good knowledge over multiple disciplines and can't really focus on one thing anymore.
Yep.
Yep, and I think you can all agree that in a PhD you have to be responsible for your own learning and that
Very
sort of
much
thing.
so.
No one will help you just because they want to.
Your lab partners aren't really lab partners. They're doing their own work, but you still kind of learn from one another. So,
Yeah, you get to do all your own pipetting
yeah.
Yeah.
and working out your own schedules.
Yes. The amount of times I've been up late, like writing up stuff to get into the lab the next morning, is just ridiculous.
Mm.
So, always good to know what you're meant to be doing. Even in the sense of, flow charts, they might seem a bit pointless, but sometimes they come in handy.
Especially if it's a new technique.
Yes.
And especially when you're trying to articulate your process to other people, like at a conference or something,
Mm-hmm. Yep.
or on a poster.
Yep.
Great poster work.
Yeah,
Cool.
Well, thank you for coming in, Vic.
no problem.
Oh, hope to see you back in the lab soon.
Yeah, I'm sure we will.
Cool.
Bye everyone.
See ya.
Oh, bye.